has to take some urgent travel. So this moment he is actually in flight. He'll reach, I think, Guwahati or Imphal. His flight is took off from Delhi at 9 a.m. IST. So he is unable to join. Vinod? Vinod?
Yeah.
Can start now?
Yeah. Yes. Okay, very good morning, all of you, and good afternoon and good evening, because many people are joined from other parts of the world. So this is the global lecture series, the 12th lecture in this series. So to begin, I request Professor Sunil Khare for the welcome address.
It's great privilege for me to welcome Dr. D. Srinivas Reddy, who is Director of CSIR IICT Hyderabad. And thank you, sir, for kindly agreeing to spend time. We all are aware you are a very distinguished scientist and Nagar Awardee and many other awards to your credit. You have done seminal contribution to this Indian science. So it's very all privileged listening from you on the topic natural products based drug delivery today. I personally and on behalf of the society feel very pleased to welcome you to this global lecture series. Thank you very much, sir.
Thank you, sir. Now I request Professor T.P. Singh for the remarks.
Yeah, this is a great pleasure for me. I was very happy to see when I received mail from you and Ashok that you invited Dr. Srinivasa Reddy. We had been interacting for a while now. And first time that we started interacting, I was in a committee and he was on the other side. Although he's such a brilliant scientist, but I could argue with him in a manner that he was not feeling very great about this. So we had together many times a lot of discussions, and subsequently, of course, his group, together with his collaborator, have synthesized many extraordinary compounds. And the fact that I made this remark with him that why he's going for natural products, because he is capable of making compounds with higher complexities. That's what I noticed. And we talked about some compounds which he was preparing for a target, and he very quickly synthesized them. So a man of his caliber, organic chemistry, I think cannot remain dependent on natural products, but I think it's interesting also to take advantage of nature's products, because whatever nature has made, or it is also part of a long experience of nature and hard efforts in many ways. So many techniques of nature eventually prepare suitable for systems in nature. They use it there and taking advantage of nature's information perhaps saves us a lot of time, and building upon that, developing compounds with better better required properties is a very good combination. So doing good organic synthesis, using nature's products and further improving them is the key to success, particularly to success in new drug discovery. And I know that Srinivas is a very highly motivated and distinguished investigator, and chemistry, particularly organic synthetic chemistry, one where we need extraordinary people if we have to succeed in drug discovery. So his many compounds he designed and synthesized in his lab. Some of them are very close to applications. So I am very happy that he's here. I'm very happy that sometimes I wonder about Ashok, how he identifies wonderful people. And this all the time makes me feel very happy about BRSI and about, I think, Ashok's, I don't know what should I say. He smells that are good people and he picks them up. And that is, I think, one of the reasons that BRSI is making progress. Really, are fasting. So, but I told Ashok that before Srinivas makes his presentation, he should be a member of the society, and then he should be. We should also consider him next time for the fellowship of society. He's such a nice scientist. Also, he has one distinction. Although he is a very hardcore lab-based dedicated scientist, at one time he was director of 3 labs. It's such a complicated situation for him, but I think he survived well. And now he's well relaxed. and he's in a lab which he likes the most. I think he's finally come to the right place. And it's a pleasure for us that he's with us today. And I thank Shok and Vinod and everybody from BRSI. We have identified him. But I thank Dr. Srinivas also that he accepted and he's with us today. Thank you very much, Srinivas. Thank you so much. And thank you, Ashok, for doing this job. All the best.
Thank you. Thank you very much, sir. Before we go to the lecture by Dr. Srinivas Reddy, I'll just give a brief introduction about Dr. Reddy. Dr. D. Srinivasa Reddy obtained PhD from University of Hyderabad, and after that he did his postdoctoral studies at the University of Chicago and the University of Kansas, USA. He has more than 20 years of research experience in total synthesis of natural products, medicinal chemistry, and drug discovery. He has 7 years of experience in pharmaceutical industries in Reddy's lab and Tata Advinus. The molecule ligoliflozin, sorry if I am wrong, discovered by his team and a team at industry is currently in phase 2 clinical trials. He has more than 120 publications and 5 patents. Dr. Reddy received several awards and recognitions, to name a few: J.C. Bose National Fellowship by Sir Shanti Swarup Bhatnagar Prize, Fellow of Indian Academy of Science, Fellow of National Academy of Science, Nasi Reliance Industries Platinum Jubilee Award, Ranbaxy Awards, and many more. With this short introduction, I request Dr. Reddy for his talk on natural product-based drug discovery. Dr. Srinivasa Reddy.
Sorry, can you hear me?
Yes, sir. Yes, sir. We are hearing you. You can share.
Okay, thank you, Dr. Vinod, Professor Ashok Pandey, Professor T.P. Singh, Professor Khare, and I see many seniors. And, I can't name all people, but I can see many seniors connected. And thank you for making me part of this BRSI talk. I heard about it, but I never got opportunity. I never got opportunity to be part of this program. And thank you for making me and giving me opportunity in this great society. So let me share my screen. Just help me. Are you able to see my screen?
Yes. It's perfect, sir. Yes. You can continue.
Okay, this today's I picked up topic natural product based drug discovery because it is very close to my heart, and also most of my research is basically around this particular topic. I started my work in Professor Mehta's lab as a natural product synthetic chemist. Then I went to University of Chicago. Then I went to Kansas, where I learned some medicinal chemistry in University of Kansas. Then I moved to industry Dr. Reddy's laboratories and Advinas Pune. There also I got some opportunity to work around this particular theme. And again, when I moved to NCL Pune, it's a similar work I did. So that's what I picked up this particular topic. And here is a outline for my today's talk. First, a little bit, since I'm heading this organization, IICT, I thought I'll use a few minutes of our this talk's time to give you a little bit about IICT. And I know many people join from different parts of the world, and I thought it will be a good information from all of you about this IICT. I know many of you are aware, but I thought students and some other colleagues who doesn't know about IICT, a little bit about, I spent some time about. Then I bring it to background on national product role in drug discovery. Then again, I talk about one case study from IIM, that is a IIM 290. And another one is next couple of projects I talk about from my own research group, that is a cladosporin, is an anti-malarial natural product. Then another one is antibacterial natural product, that is a funanimycin. Then in the end, I'll try to give some message from this talk. So we are part of a big organization called Council of Scientific and Industrial Research. This is one of the biggest organizations in our country. And we have about 37 laboratories across India, starting from Jammu Kashmir to Trivandrum. And we work in different domains of science, starting from physical sciences, chemical sciences, information sciences, engineering sciences, and biological sciences. So our president is always Prime Minister of India, Vice President is always Minister of Science and Technology. So recently we have new Director General and first woman DG after almost 8 decades of our existence, Dr. Kalai Selvi. She came from a small place in Tamil Nadu, Karaikudi lab she was heading before, and we are fortunate to have her here, very dynamic leader, what we have here. And my journey started in CSIR in National Chemical Laboratory, Pune. So I was working in Pune at that time with Tata Advinas. There was one advertisement from NCL Pune. So I applied, then I got into NCL Pune in the year 2010. So then later, I spent almost 10 years. Fortunately, I got good students and we did reasonably good work. Then after spending about 10 years in National Chemical Laboratory Pune, then I moved to northern part of our country, that is Jammu Kashmir, where I was heading Indian Institute of Integrative Medicine for a couple of years. And during that time, I also got opportunity to have additional charge of CDRI, that is Central Drug Research Institute in Lucknow. And in the last year, when Dr. Chandrasekhar of IICT director at that time, when he was elevated to DST secretary, this position fell vacant of IICT director. Then there was advertisement, then I applied. Then that's how I am here in IICT. So that's basically, I got a rare opportunity for any scientist to work in different labs. I was lucky to get opportunity to work in different laboratories of different geographical regions. So IICT established before independence, that is in 1944. That time it used to be CLSIR. Then later it was renamed to RRL Hyderabad. That is a well-known RRL Hyderabad. We have few labs across country like RRL Jorhat, RRL different parts of the country we have, like 3-4 labs we have. So recently, not recently, in the year 1989, this RRL Hyderabad renamed to IICT, that is the Indian Institute of Chemical Technology. Our vision is to serve society by creating an outstanding knowledge base in chemistry and chemical technology. That's what we look forward in our institute. We have different departments, although our main core area is chemistry and chemical technology, but we work in different fields of, we have different departments, and starting from organic synthesis process chemistry, and we have natural products and medicinal chemistry, catalysis, fine chemicals, energy, environmental, and we have good presence in oils and lipid science technology. And we have one of the well-known department, analytical and structural chemistry, and we have applied biology, process engineering, tech transfers, and fluoroorganics and polymer functional materials. So basically, we work with all this in different departments, but again, ultimately, we work in four major problems, that is healthcare, chemicals, energy, and environment. These four, mainly we surrounded around these four themes: healthcare, chemicals, energy, and environment. So we are located in a place, in a city. It's like a very attractive place. It's basically presence of many academic institutions and industry presence in particularly in pharma industry is well known in this city. And we have in our institute, we have about 150 plus scientists, and we have 300 plus technical support, and we have about 500 plus research fellows always will be floating in our institute. So our institute contributed in many areas, starting from coal in the very beginning, and fine chemicals, pharmaceuticals, lipids, agriculture, waste to wealth. And very recently, we got into the circular economy, biohydrogens, chemical biology, catalysis, and we supported many industries from concept to commercialization. There are many, many significant achievements made by our institute. Again, I'm thankful to my management of CSIR giving me this opportunity to head this organization. And we have contributed many ways, but I'll tell you in recent pandemic, I'm just giving you a couple of examples of recent contributions from our institute. So as all we know, we were struggling during COVID pandemic. IICT and many other CSIR labs got together, and we worked in many aspects of many aspects in COVID pandemic situation to address those issues. Particularly CSIR, IICT worked on process development of a couple of molecules. One is with the favipiravir, another one is remdesivir. Favipiravir technology was transferred to Cipla in collaboration. They further developed, and it is now marketed as Ciplenza. So there's a big contribution during that time. And remdesivir also, we worked quite a bit on this one. And more than that, another big contribution is adjuvant, particularly in Bharat Biotech's Covaxin. So our IICT developed a process, so very cost-effective, scalable process developed in our lab at IICT. That is a toll-like receptor 7/8 agonist molecule. And particularly during the tough times with available chemicals, they worked together with Bharat Biotech, and that's what resulted in Covaxin shot. And I'm sure many of you and took this Covaxin shot, where we have little contribution, microgram levels of our adjuvant is present in that Covaxin. That's another big contribution from our institute during COVID pandemic situation. I also had, at that time, I was heading IIM Jammu. That time we started repurposing of old drugs, particularly colchicine. Again, this is one other right example for this particular talk. It's a natural product became drug, that is a colchicine. So colchicine along another one is a niclosamide. These two compounds were tried for clinical trials. That time we were closely working with the IIM, LuxAI, and IICT together. So we have completed our clinical trials. Now it is presently in the evaluation stage. Hopefully, if everything goes right, all the data, at least data is promising at this moment. We are planning to approach DCGI with this data to get approval. So that's what our contributions during a pandemic. And just for the benefit of students, how do you get into our institutes? Not only IICT, any another institute. If you want to join in a PhD program in different disciplines, you need to have a master's degree with valid fellowship. It can be CSIR or UGC or ICMR or Inspire Fellowship. You can also join as a scientist. If you are a PhD or PhD plus postdoc experience or industry experience, like me, after spending industry about 7 years, I joined as a scientist in INCL Pune. So like that, anybody can join here. You can also join as a project assistant or associate if you have MSc, MPharm, or PhD or experience. So these are all temporary positions also one can join here. Okay, coming back to talk of today's talk. So key milestones in 19th century is, again, there are many molecules, but I picked a few molecules which made real impact. That is, if you look at this very early 1800, there's a morphine. So that was originally isolated in 1806, but later its structure was illustrated by Robinson in 1925, and synthesis by Ginsberg in 1954, and biogenesis also later from Leite from 1959. You look at this one, at that time we didn't have enough tools, but they could establish all the structures and synthesis at that time. And in 1818, strychnine was isolated. Again, 1946, it took almost more than 100 years to establish its structure. Again, synthesis was well known. That is 1954 by Woodward, and later it was also done by 2001 in Berg, and many other people also synthesized later. And another important molecule, landmark, is quinine. It was isolated in 1820, and synthesis was 1944 by Woodward. Again, you look at this, the top three molecules, these are all done much before our Wohler's synthesis of urea. So that's kind of like contributions our chemistry world made to the society, like to the world. And camphor, cocaine, and quinine, these are all other natural product. It was isolated in later part of 19th century. So why natural products are important? So just again, I'm sure some of you are already aware, but just to have justification. So more than 40% of the today's marketed drugs are related to natural products. So there is no doubt about it, whether directly or indirectly, they are all connected to natural products. Close to half of drugs what available today in the market. So they have special features. That's where they are becoming very useful in discovering drugs, particularly scaffold diversity. The kind of scaffold diversity you don't get in synthetic molecules. So that's a big advantage. So you don't know which kind of scaffold you get from natural products. So difficult to predict in the beginning. And structural complexity, particularly three-dimensional structures, that is very, very important. And particularly if you want to go for structure-based drug discovery, and if you want to have a proper systematic way of, if you want to do research and like drug discovery, this is one of the way you can go forward. If three-dimensional structures are available, that is the best way to go forward. And they typically have higher molecular mass and larger number of sp3 carbon atoms. Again, that's what I was telling you, is more stereo centers. That's the advantage with these natural products. Larger number of oxygens and fewer nitrogen and halogens. Higher number of hydrogen bond acceptors and donors. Of course, because of this, actually, we also sometimes we move away from our rules, that Lipinski rule of 5. So there are some exceptions, many exceptions in natural products, in natural product based drugs. So lower C log piece, that means more attracted to water. There is a higher hydrophilicity. That's the advantage with the natural products. And greater molecular rigidity compared with the synthetic molecules. Again, these are all because of three-dimensional structures. Most of the synthetic molecules are, in general, I can tell you that they're all heterocycles. They are all more aromatic structures, more flat molecules. So because of this molecular rigidity compared to synthetic compounds, this will become more attractive for discovering like in a rational manner. So natural products based drug discovery, there are always challenges. If you look at this, our traditional approach, starting from organisms, it can be anything. You can also crude extract you take, and you make fractions, multiple round of fractionation you do, and then based on bioactivity evaluation, then finally you get into isolation of single pure compound. That's a general practice we have been practicing. But again, most of the time we hear from different people that different problems. Again, look at this problem one, like not possible to culture organisms out of the natural habitat. This has been a big problem. But if you look at new methods, recent methods, they're all helping us in culturing those organisms even out of the natural habitat. That's again another advantage in recent times. Similarly, relevant natural products are not produced when the organism taken out of the natural. Again, it is basically very similar to that one. We have now introduction of this heterologous expression of biosynthetic genes. These are all again new techniques helping us. Even if you look at this problem 3, 4, 5, where presence of natural products are not known. Again, these are all problems, but now there are new methods are available, even finding compounds. Even if it is in a minute quantity, we are able to do it, and we are able to fractionate, we're able to do isolate small number of, like small quantities of the natural products. So that's the advantage what we have here. And problem 6 is like unknown molecular targets for natural products identified with phenotypic assays. Again, this problem with even synthetic compounds also comes, but again, there are new methods are available and mode of action in illustration of structures. They're all again available modern techniques. That's where we need to combine our traditional knowledge with modern science. Then that's the way to go forward. That's what we have been doing. There's one beautiful review written in Nature Reviews in Drug Discovery. If you are interested in this field, this is one of the recent review and well cited and with the good information available in that. Natural products, human medicine. Again, I'm giving you a few examples which made impact on our lives. Penicillin, it's antibiotic. I'm sure I don't have to tell you about how penicillin saved our world. Taxol, anti-cancer compound. Then CNS, basically this another compound, reserpine, artemisinin, vinblastine, FK506. These are all again, it's made our life, today's lives easy because of without these molecules, imagine our lives or imagine our world. Look at, I'll give you one example, this FK506 is an immunomodulator. We can't think of today's organ transplant without these immunomodulators. This is again, these are all nature's gifts for us, for all of us. So just to give you a little more information about reserpine, this is it has Indian connection. It is isolated from Sarpagandha. Its scientific name is Rauwolfia serpentina. So it isolated from the dried roots of that particular species, this particular plant. And there are masterminds behind this one, particularly R.N. Chopra and Professor Mukherjee, APC Ray, and Dr. Gannath Sen, is K.C. Bose, and Dr. Siddiqui. So these are all, many people worked on this molecules and contributed the extensive development of this reserpine as a medicine. So I was fortunate to see this Rauwolfia serpentina plants in our IIM farms. So that's where when I went there, I took picture. This is in our on our own farms, CSR farms. This is, and also another connection I have with this reserpine is I worked on synthesis of this particular molecule during my PhD. I spent almost 3 to 4 years just to make this compound. That's another connection I have with this reserpine. So in India, if you want to do natural product based drug discovery, I can tell you IIM, that is Indian Institute of Integrative Medicine, is one of the best place because everything is under one roof, and they have fantastic facilities for plant sciences, microbiology, and basically that's where you isolate initial compounds, natural products. Then you have fantastic pharmacology. Then from there you have informatics, medicinal chemistry, and development, and you also have fantastic facility of fermentation technology. We have all other facilities required for discovering drugs. So that is where I was lucky to be part of that organization for some time, and also that's where my interest basically increased more when I went there. So that's IIM. I am sure if you are, some of you may be aware, but if you are not aware, I think I request you to visit IIM, either virtually or physically. You'll get to know more about that particular institute. So that institute also developed a library. So it is stored this is a repository. So we have NC is about 2000, and natural products are pure natural products are about 600, and also externally produced about 80,000, almost about close to 85,000 compound libraries available. And particularly more interesting is natural products available, pure natural products. I don't think any other place this pure natural product library is available. And if you are interested, you can write to M director, then they'll get connected to concerned scientists. So they also basically screen into many assays, and there are several hits they found: tuberculosis, antibacterial, also different targets also as part of antibacterial, and Alzheimer's disease. And there are also PDE5 inhibitors, Notch inhibitors, Notch activators, PGP inhibitors. There are also several anti-cancer molecules. There are also few oral care molecules also they found in their library. So what I mean to say, like, this is one of the place where you can find pure natural product stored in their facility. And I'm sure they'll be happy to collaborate with in different parts of our country and also world. And here is a snapshot of some molecules, different natural products being stored at IIIM. And these are only a few examples, but there are different scaffolds are available in that library. So this is another contribution, natural product based drug discovery. I cannot call it as a new drug discovery. This is something interesting. There is a scientific basis for discovering this rhizorin. Rhizorin, it is actually connection came from Ayurveda side. Other way, there is a Trika-2. There is a basically 3 component mixture. That is a Piper longum, Piper nigrum, and Gingiber officinalis. Basically, it is a long pepper, black pepper, and ginger. The combination of these 3. So that gives Trika-2. So that's where original idea came from there, where it was increasing bioavailability. Then later they isolated a compound called piperin from Piper species. So that improved bioavailability of some of the anti-tuberculosis drug. So that's where originally it started. And we have a combination drug that is a piperin rifampicin combination, where rifampicin used about 200 mg and piperin used 10 mg, which is equivalent to bioequivalent of 450 mg, almost like half of the quantity reduced rifampicin. So that's where similar effect is giving. So that is one of the, I can tell you, this is one example we can give where traditional knowledge used for discovering new drugs. Although it is not a new drugs, but a new FDC, that is a fixed dose combination, I can tell you. So this, there are also scientific rationale is there. Later they found that it inhibits CYP3A4 mediated phase 1 metabolism in liver and intestine, and also it inhibits PGP mediated efflux. So basically, accordingly, this increased absorption permeation across like GIT. So it was marketed in India in 2009 in public-private partnership with Cadila Pharmaceuticals. That's again one of the contributions from natural product-based drug discovery, in particularly using traditional knowledge in medicines. Here is another example. From IIIM, one case study. This is IIIM 290. It was originally generated from a plant, Indian medicinal plant, rohithikin. Then rohithikin turned into IIIM 290. That's currently they are planning to take it into clinical trials. That is phase 1, phase 2 clinical trials. Just I'll give you a little bit more information about this particular story, this particular IIIM 290 story. Look at this plant. This is a very big tree. This comes from Miliaceae, and in India it is called Indian white cedar or Lasuni amari. So that's basically, it's a very huge tree. Mainly they use it for wood purpose and with smooth surface and building construction boxes and many like basically it's kind of a wood material. We can use it for many things, even cigar boxes and plyboard also they use. And in India, it is primarily available in Western Ghats, that in Karnataka, and also in Dehradun, this Forest Research Institute campus mainly. So these two places mainly it is available. In other parts of the country also it is available. It is a rich source of rohithikin. It's a chromone alkaloid. So in 90s, our Indian chemists isolated rohithikin from this disoxylum Binactyophyrum hook, which is again phylogenetically related to this Ayurvedic plant, again another species of disoxylum, which was used in rheumatoid arthritis. So that's the background, and rohithikin was isolated as a, and also it is responsible for anti-inflammatory and and also immunomodulatory activity. So that's another interesting thing about this is this product, although initially they did from bark and other parts of the plant, but later they found that in leaves, which are renewable sources. So from leaves, they could isolate this particular rohithikin, and it is also available in fruits also. About 0.5 to 5%. It ranges from depending on the location where you get your leaves. So disoxylum leaves, one can isolate rohithikin. So rohithikin is a starting point for two drugs which are under clinical development. That is flavopiridol, that is Sanofi, which is being developed as an orphan drug in phase 2 in numerous cancers. Another molecule is P27600. That is in India, Piramal. It's being developed under in India and phase 2 head and neck cancers. So that's kind of background. Without background, IIIM. Before that, actually, there is a lot of SAR is available in the public domain. So look at the structure of this particular molecule, rohithikin. So they divided into 4 parts, A, B, C, D. Again, in A, because of phenolic group, you can do etherification, and B, there you can introduce manic products and Bale-Silman products, and also Claisen-Schmidt condensation also they used for making other D functionalization at D position. So that's what they used some of the common reactions to make a variety of compounds around rohithikin. So that's kind of starting point, and also they looked at other interactions. This is a CDK9 cyclin T1 binding site, and they found that there are some interesting interactions. So they designed these molecules. So based on synthetic chemist experience, they made several compounds, particularly this sterile derivatives, we call it. And several compounds, easy to make if you have corresponding aldehyde, and if you by looking, you can make many compounds. There are few examples. There are few examples here. I'm showing you here. One particular compound is a 2,6-dichlorophenyl is there, corresponding aldehyde is made, and that particular compound became lead, and later it became as a front runner in this whole series. So here is information about some of the xenograph efficacy studies in MIAPAC calcium 2 xenograph model. This is one of the model used for these cancers. So dose dependent tumor growth inhibition, and also 51 TGI up to 51% achieved, and no mortality in any of the dose. That is interesting. Generally, cancer medicines you have a less window, and particularly this compound seems to be seems to have a good index, safety index, and lowest tested dose was 15 mg per kg. It's also efficacious. So you can see the beautiful dose response in xenograph model. And other parameters also completed. If you want to take forward molecule to clinical trials, this IAM 290, all this target identification. So NCE patent also there, formulation patent also very recently it is filed. And we got now DCGI grant, DCGI granted permission for conducting clinical trials and it will be soon started. And we are in the process of making GMP material for this particular compound. And we all, basically there are list is here, and efficacy established in animal models and ADME properties, safety pharmacology. Particularly, we all completed this HARG, mutagenicity, genotoxicity, acute, subacute tax, all completed, and CMC documentation also complete, and patent also filed and granted in US patent also. So this is what one of the successful story I can talk about it proudly. We can talk about proudly in India. We started from plants, and we start natural product was isolated, that rohitkin, then later converted into IIIM 290 by synthetic chemistry expertise and medicinal chemistry combination. Along with the biologists, they discovered this drug IIIM 290. Particularly Dr. Sandeep Barate and Dr. Ram Vishwakarma was main players for this program. I was happy to know that things are moving, and we are going to do clinical trials using this IIIM 290. Now coming back to some of my work done in my research group. After spending 7 years in industry, when I moved to MCL Pune, I had a choice to work on medicinal chemistry, where I learned in industry, and also natural product synthesis, where I got training in my PhD and postdoc. So considering the importance of both fields, I started both natural product synthesis and also medicinal chemistry. In addition to that, I also believe that way forward for future is application-oriented chemistry. Particularly since I know our group is interested in organic synthesis, we took up theme application-oriented organic synthesis. We strongly believe in collaborations. Although we make only molecules, but we can get connected with different parts of country and also world. We get connected. We try to create value for the molecules what we make in our laboratory. So in particularly, our medicinal chemistry programs move around antibacterial, antiparasitic, antidiabetic, and anti-inflammatory. And particularly, why are you interested? I know I gave a justification why natural products are important. I gave that justification, but why natural product synthesis is important? Because 80% of the commercial drug today derived from natural product required synthetic effects. Although I said about 40% are linked with natural product, but among that 80% required synthetic efforts, because most of the time these natural product as such, they don't become drugs. They need to be further tuned, because most of the time their solubility or toxicity or selectivity, those are the problems. Unless you have synthetic modification, you will not be able to do it. In addition to that, if you want to make more quantities, isolation may not be the right choice for all the time, because you need to make in good quantities, large quantities. Further synthetic efforts are required. So in addition to that, personally, I believe that that students who work in total synthesis, they get exposure to variety of reactions. So make them strong scientifically and also mentally. That's what personally I believe. This total synthesis offers structural modification, structural simplification, and also supply of natural product. That's a background before taking up total synthesis. I'll give you one more example from my industry experience. Here is a natural product called fluorescein. So this was showed this particular natural product fluorescein showed anti-diabetic effects. Of course, in the beginning they didn't know about the target, but later they found that this is working through sodium glucose co-transporter inhibition. So later structural modification around this molecule were taken up from different groups. Particularly, I think Japanese scientists worked on this, and they made a compound called T1095. Again, that particular compound also has selectivity issues and also stability. You look at this compound, this is called O-glycoside. So our body has glycosidase enzymes. So these glycosidase enzymes can cleave this O-glycosidic bond. So to address those issues, then groups across the world came up with the compounds called C-glycosides. And finally, that is again, that compound is stable for glycosidase enzymes and also selective for particular subtype. So dapagliflozin was discovered, and later panagliflozin, and there are many other, almost like there are 5-6 already in the pipeline of different glyphosins. Again, they're all worked through SGLT inhibitions. And during that time, when I was working in Advinus, along with the Novartis collaboration, we discovered a compound called LIK066. So now it is called licoglyphosin. So where I was project leader for that particular program. Again, this is again big satisfaction for me with the help of group. It's a big group we're working. The help of chemists, we made this compound. Finally, it is in human clinical trials, and it is also being evaluated for other indications like NASH and PCO patients. So here is when I started my research at NCL Pune, our goal was to make natural products with which have biological activity, then take them forward towards medicinal chemistry, and finally come up with the leads. That was a bigger theme. Ultimately, discovering drugs from natural product. That was the idea. So we picked up natural products from different scaffolds. Our main goal was to pick up natural products which are suitable for synthesis and modulation of their structures, so that it is easy to modify and make them as much as possible libraries. So that's the idea behind picking up some of these natural products. So look at this first example, peribacin D, that the cell addition inhibitors that can be used for towards cancers or sickle cell anemia and few other indications one can think of, and anti-inflammation also you can use. And also this other molecules like anti-diabetic, anti-cancer, anti-inflammatory, and anti-malarial, antibacterial. There are different scaffolds. This again, here I'll give you one example. This solomonomide, it's anti-inflammatory compound. This is one of that project we have been working for almost more than a decade now, and it gave us 2 SCRB grants and got 2 PhDs and several papers just based on one natural product. That was so interesting molecules, but I'm not going to talk about that particular natural product today. So today I'm going to spend time on 2 natural products, that is a cladosporin and hunanimicin. How we pick up and how we do in our lab. This is what I thought I will showcase using these 2 examples. So first case is cladosporin. It's a potent anti-malarial compound. It was isolated more than 50 years ago. In 1971, it was isolated and from fungus. And but why are we interested? It's an old molecule because recently it showed basically it was reported recently that it is working through lysyl tRNA synthetase of Plasmodium falciparum, which inhibits protein translation in the parasites. That was a recent discovery a few years ago. Where and also in addition to that, it is also selective with the human enzyme. So these are the advantages. So that is a very potent nanomolar antimalarial activity and selectivity made us to pick up this particular natural product for our program. And if you look at this particular compound, although the synthesis was already reported a few years ago, particularly Shi et al. In 2012, and one of my colleagues who is presently at IICT, Mahapatra, reported synthesis 2013. Here, when we pick up this particular molecule, our interest was to do something different, systematic SAR. Look at this molecule. There are 3 chiral centers, 1, 2, 3 chiral centers. That means 2 to the power of 3, 8 compounds are possible. We wanted to understand which particular configuration is suitable for moving forward, or which 3-dimensional structure is suitable for moving forward. That's what the idea when we started. For that purpose, you need to make all the 8 possible isomers. So during that time, one of the student from IIT Madras, Mr. Pranay, joined in my group. So we discussed and we came up with this plan. Let's make all 8 possible isomers, then subject them to biological assays using enzymatic and parasitic assays, and finally come up with the active stereochemical arrangement and for further modifications towards potent lead. That was the idea when we started this program. So accordingly, we started with dimethoxybromobenzene, and with few steps again, since most of the audience here are from biology background, I keep minimum synthesis here. This allyl compound in fragment A we made, and similarly starting from S-propylene oxide, we made fragment B. Once we have fragment A and fragment B, we combined both using a reaction called Grubbs metathesis. Then you get these two compounds, then you separate both of them, then both of them independently take them to with using some of the organic chemistry experience, and you make a compound clado-1, clado-2, and similarly other isomer will give clado-3, clado-4. We made 4 compounds using one particular epoxide. Similarly, if you use R epoxide, you get other set of 4 isomers. Like that, you get clado-5, clado-6, clado-7, clado-8. Another challenging part in this particular project is characterization of all the compounds, because they are very closely related. It's difficult to distinguish with each other. So although we have some logic and some of the NMR studies we used to find out exact configuration, but ultimately we got a solid proof with crystal structures. So we were able to crystallize all the 4 compounds. We were lucky to get all the crystal structures, and we found that whatever we assigned were the right structure. Once we make these compounds, once we made all these compounds in collaboration with Dr. Amit Sharma and Dr. Dhanashekaran Shanmugam of CSR NCL, we collaborated with them because they are biologists who work on this anti-malaria projects. So all the library compound subjected to TSA, that is thermal shift assay, followed by parasite growth inhibition assay, and followed by enzymatic inhibition assay, and finally find out active stereoisomer. So accordingly, what we did is we also co-crystallized all the compounds. So fortunately, we got only 2 co-crystals with the 2 compounds. Out of 8 compounds, only 2 compounds are binding with the lysyl tRNA. We got those 2 crystal structure, and finally we found that only those 2 are the compounds are most potent compounds. And that's where we found that out of 8 compounds, only 2 compounds. This is particularly 3R10R14S, and another one is 3R10R14R. The difference between both the most active compounds are on 14S configuration. That is this methyl stereochemistry. So methyl, that means methyl stereochemistry doesn't matter whether it is R or S, but this other 2 centers are very, very important. That's where we found from this study. So entire stereochemistry, stereochemical set of compounds were studied, and we found that these 2 compounds are important. Other, of course, there are class B, class C also we found, but I think for our purpose, class A became the best compounds. And this is not the first time we are showing, but using this particular scaffold and using this particular lysyl tRNA synthesis, we showed that stereochemistry is very, very important in drug discovery. So once you have this one, then what next? Then we have to make a library of compounds around this compound to further optimize towards lead compound. So we picked up different regions like dihydrocoumarin moiety and tetrahydropyrin moiety and linker. So these are the picked up with different moieties, like different regions. Accordingly, we made library of compounds. We made about 28 compounds, analogs, and found that some of the compounds are showing interesting, and there are some of the compounds also pharmacokinetically also some of the compounds are showing interesting properties. So this again, we went through this all the screening cascade, and finally we have about 8 active compounds. And finally, what I'm going to show you is during the lead optimization of original natural product. So through structure activity relationship, we have now lead molecule, we call it as a CL2. If you look at the difference between these 2 compounds, only the introduction of, although we did a lot of modifications and functionalization around this scaffold. So ultimately, we found that introduction of this hydroxy group in a particular manner is only going to be our future lead. So this is what, again, we got co-crystal structure with the help of Dr. Amit Sharma of ICGB, and recently published in Asia's infectious diseases. Now we are moving forward with this molecule. So what we found is during the lead optimization, again, I want to give information that just not the potency is not important, other druggable properties also important. Here, if you look at what is happening is from when we move from cladosporine to that lead presented that Cl2. So metabolic stability improved, and aqueous solubility improved, and the number of hydrogen bonds also improved. So basically that acceptor also improved. So these are all things can give us more druggable properties. Basically drug likeness improved when we moved from cladosporine to Cl2. That's what we wanted to show it and also establish this proof. So this is what we discovered now, lead compound with the systematic SAR. And now on last story I'm going to tell is hunanimycin story. This is antibacterial natural product. It showed activity against Salmonella enterica, which causes food poisoning. Although we don't have a serious, at least we don't have data in our India, but I can tell you that this global burden of foodborne disease is about 600 million foodborne illness and 4.2 lakh deaths occur annually. It poses more risk in low income and sub-regions of the world. So that's basically that's a background. And look at the structure. It's a marine natural product from marine organism. It was isolated and possessing salmonella activity, anti-salmonella activity, and it is very similar to riboflavin. So the difference, the sugar moiety is same because ribose, and the only thing is skeleton is a linear skeleton. Here is a angular skeleton. That's the difference. And when we calculated Lipinski rule of 5 for this molecule, so there is no violation. So basically perfectly is a fitting with like aligning with our Lipinski rule of 5. So that's where, although we don't use Lipinski rule of 5 all the time, in each molecule, but wherever Lipinski rule of applying and which is where there is no violation, we pick up those molecules as a priority. So hunanimycin we picked up, and there was no synthesis available, only less than a milligram it was isolated. So we thought we'll come up with a scalable synthesis and focus library and lead optimization using this scaffold. That was the idea when we started. So it's easy to make. It's a starting material. If you have corresponding diamine, treat with oxalic acid or diethyl oxalate, you get solid crashes out. Then you do monoalkylation, and followed by Friedel-Crafts alkylation gives us this tricyclic compound. Once you have this tricyclic compound, so next job is to install this ribose moiety. For that purpose, we used one of the allyl derivative, then followed by osmium tetroxide mediated dihydroxylation, and followed by deprotection of this estonide gave us hunanimycin. Originally, this compound was isolated by that group less than a milligram. So now we prepared more than 80 milligrams. It was published some time ago. And later, we thought that as a synthetic chemist, we were not happy with the synthesis. Look at the starting material cost. Diamine, although looking very simple molecule, it cost is about 25 grams of cost, $450. And also it takes about 11 steps with 7% yield to get to hunanimycin. Then we thought that probably, why can't we start making from riboflavin? Probably nature is making from riboflavin. When you look at the cost, very cheap, that is $32 for of the same quantity of 25 grams. So then we start with the student and we came up with a scheme. Why don't we start with this riboflavin scheme? So accordingly, what we did is we use riboflavin as a starting material, which you can buy, bulk quantities you can buy. And sodium hydroxide in presence of hydrogen peroxide gave us this compound where this ring is removed. Basically, this got hydrolyzed to get to this compound. So once you have this particular compound, then you can do prenylation using potassium carbonate. You get to this N-alkylated product. And finally, Friedel-Crafts alkylation gave us hunanimycin. Look at this one. It's a beautiful synthesis. 3-step sequence. You can make gram scale, no column purification, biomimetic approach. Overall, this is close to 50%, 90%, 96% purity by HPLC. So original isolation is 0.8 milligram. Now we can make multiple grams of compound, this compound. So anybody can, it's so simple synthesis we developed. Any undergraduate student also can make this compound within no time because riboflavin is available plenty and some of the reagents also available on benchtop easily. So anybody can make this compound. So now what next? We made No, we also wanted to understand whether it is going, what mechanism it is going through. This is something interesting. I thought I'll share with you since we have a lot of biologists here. Because of the structural similarity, we thought that it is going through riboflavin synthase inhibition. So because of structural similarity, then we wanted to establish through mechanism. For that purpose, we collaborated with Macmillan of University of California, Santa Cruz. So what that group did is they incubated in E. coli in presence of riboflavin synthase with a decreasing concentration, increase the production of riboflavin. So they basically inversely proportional. So that means it is indeed is going through riboflavin synthase inhibition. So that's kind of we have a full support, mechanistic support we got. Of course, we ultimately would like to go for co-crystal structure and come up with a like a more confirmed way, but this is what at least is giving indication for us that it is going through riboflavin synthase inhibition. This is something interesting target because riboflavin synthase is not there in humans or higher species. This is only present in some of the bacteria. So if you make drugs against this target, it can selectively kill the bacteria, sparing all humans and higher species. That is the advantage with this particular target. We get riboflavin from food, whereas bacteria they synthesize using this riboflavin synthase enzyme. So that's the difference between humans and bacteria. So later, what we did is, as part of our medicinal chemistry program, we came up with the library of around these compounds. Again, not going to the synthesis of these molecules, but finally what we did is we found a molecule 100548 is 1 microgram per ml. Original molecule showed in our hands 8 microgram per ml MIC. It's almost 8-fold under the same condition, 8-fold improvement. And these some of the parameters we found using SAR. And we have synthesized more than 70 compounds, and all of them are screened against Salmonella enterica. And we have 8-fold potent compound and mechanism of action established. Although ultimately we look forward for co-crystal structure, but at least we have some indication that it is going through riboflavin synthase inhibition. And lead compound showed excellent in vitro PK profile, pharmacokinetic profiles. And also we found, we have more than 2 gram per kg LD50. So that means it is a safe molecule. So we are doing further in vivo studies in collaboration with the Professor Macmillan. And also we are thinking that why can't we use this molecule as a food preservative, and it need to be explored. It has a huge market potential. So we have some of the data also generated, particularly pharmacokinetic data generated from Incogen Therapeutics of Hyderabad. And what I try to show last 30-40 minutes is we work on application-oriented organic synthesis, in particular on natural product synthesis towards application of human welfare, in particular addressing human health problems. So we have identified few leads, and hopefully they're going to be useful in the near future. And more than that, students who trained in my research group, at least they'll be oriented towards application-oriented organic synthesis, particularly in medicinal chemistry using natural product scaffold. So what is that I'm trying to give message here is, although there are challenges, natural product based drug discovery is going to play an important role in our future discoveries, and it will provide contributions for human health. Of course, more systematic efforts are required, particularly recent advances in biosynthetic engineering technologies, and also screening libraries with natural product hybrids, analogs, and there are also compounds coming in natural product inspired molecules. There is also in chemistry, I can tell you that late stage functionalization is also very important, and that is also giving us a new set of libraries. A natural product based drug discovery, I can also tell that this is a unique platform for different collaborations. It can be academic or international collaborations. I think both chemists and biologists together can discover drugs from natural product that can lead to future drugs. That's what I'm trying to give. Natural product field is really interesting, and I encourage all people to work in that direction. So here is my research group and current group. Last couple of years, we got little scattered between Pune, Jammu, Hyderabad. Now we all are together in Hyderabad, and this is my current research group, and all other members who graduated from my research group who are outside that frame. So they are all graduated, and some of them are faculty, and some of them are industry, and outside they are there. And finally, I would like to thank all my funding agencies and collaborators. I think collaborators, I was lucky to get good collaborators from different organizations in India and outside. Although we know only we make only molecules, but without collaboration, without support from the biology group in particularly, I don't think we would have created values for the molecules what we made. And thank you once again for giving me this opportunity to show some of our work. Thank you. Thank you very much.
Oh, sure. We can ask some questions.
Yes, sir.
Yes, please.
Questions
Thank you,
and also
Srinivas. It was a very, informative talk, and now the forum is open for any question, short questions and comments, which I am sure Dr. Reddy will be happy to take. Processing.
yes, sir. Questions and also suggestions also I'll take.
Okay. So maybe I can begin. I think this is an extraordinary talk, Dr. Reddy. That talk is very extraordinary and the work is so fantastic. And so many things you have done. My one fundamental question with you is this. You talk about natural products. Now, these are plant metabolites. These compounds, are they utilized by plants to bind to some targets in plants? If that is the case, that would be interesting to know which kind of protein targets they bind. So we can see similarity in the human proteins with them to correlate that it could be a useful compound to be used in humans. Or number two, another important point, your plant products have extraordinary chemistry. They have very exciting chemical structures, which we still haven't, in the synthetic lab, we still haven't made, or let's say they are different than what you make in the lab. So one is that, do you use these products primarily to take advantage of the chemistry carried out in plants, and you want to make this chemistry basis and build on that the potent molecules for your applications? These are the two things you can comment on. Thank you so much in advance.
Thank you, sir. Thank you.
Now, my question is over. Now it's for you to comment.
No, I thought you made comments, sir. What is that question you exactly asked?
My question is the compounds you isolate from plants.
Right.
Are they utilized by plants for something? Or they are just there as a metabolic products and not used by plants? Number one. Number two, what is your goal? You want to simply use the chemistry of plants to build upon the framework, make molecules for yourself, for your applications, or you use them directly for some biological applications. And if you use them for biological application, what is your logic to use them? Trial and error could be one thing, but is there any scientific basis in this?
Yes. So answer to first question is, in a plant, I'm sure there must be some reason. That is why these compounds are made as part of biosynthetic pathways. Again, we know that one, they're all important. And not only that, even not just plants, even endophytes. So some of them also, they have reason to protect plants for different reasons. I think these natural products are originally made, but we are using compounds made in plants or any other organism for human purpose. So I'm sure there must be some kind of common pathways or common use both in plants and humans or animals. I think there must be. I don't think anybody did systematic, but there are some papers I see that common pathways in plants and animals. So that could be one different way one can look at. But as such, I don't think it. Sometimes it need not be the same purpose. It can be a different purpose. Sometimes some of the targets which are expressed in plants and animals also is a different. So that way also it can be useful. But coming back to second question, and yes, sir, we are going to use these compounds for mainly for human use, and we are doing some systematic approach. Although we use a chemistry approach, ultimately we connect with structural biologists like you, and we come up with the rational drug design to move forward. That is one aspect. But another aspect is we create libraries of molecules. Need not be that I have to use. Anybody else also can use this, pick up these molecules, and they can use it for their own purpose. So that's what I can tell.
But it would be nice to find out the potential targets in plants. It can help us to identify, correlate with the targets humans. That may be.
I'm sure there must be some people must be doing, particularly people like you can help actually, finding common target between plants and animals.
Okay, thank you very much for the nice presentation and nice answer to the question. All the best.
Thank you, sir.
Ashok, I'm okay.
Okay, thank you. Anybody else has a question? Renu, you have something?
Yes. If I can ask. Excellent talk,
Yes,
Dr.
please.
Reddy. Very impressive chemistry. My question comes from my background as a molecular biologist. Most of the time, what we find is that the individual purified components do not work so well, and the extract or a mixture works better. So when you, in your terminology, when you are doing this natural products, you're end of the day, you are still looking at making them drugs, and you're looking at drug chemistry or drug bioavailability. My question is for you to comment on how would you take the plant extracts or combinations to be working? Do you think they are better, or you still would like to have the single targeted approach and do your amazing chemistry on them?
Thank you for the question. And yes, you need to, in some cases, definitely you need to have synergy. You need to have multiple components, particularly if you look at our traditional medicine, like Ayurveda medicine.
Yes.
I think unless you have that components, you will not be able to see the effects. But at least my approach is take information from there, that traditional knowledge, use that plant, particularly what Rohit Kinu isolated from this disoxylum. So that's originally it was that plant being used for anti-inflammatory effects, some of those things. That's where we picked up the knowledge. But later, ultimately, you need to come up with the rational drug design. If you want to take it in a DCGA or FDA approved drugs, modern drugs, I think you need to go through this a single compound, purified compound, find a like a target and establish all the possible basically biological profile, including toxicity and everything, and move forward. Ultimately, that will give more acceptable from all over the world. Although we some prefer Ayush based drugs, it can be Ayurveda or Siddha or homeopathy, any other place, but we can take that knowledge and come up with the new molecules. But again, is there also combination? If you look at, there is a combination of drugs, there's fixed dose combinations. That is also partly related to extracts. So probably you need some synergistic effect from other components as well. But as far as I am concerned, I prefer going with a single molecule with a systematic approach. That's my preference, but we always take our traditional knowledge information.
Thank you. Thank you so much. There is something called botanical drug as well, which is an upcoming field in FDA as well. So I
Yes.
would really request the Indian community, Ayurveda, we all promote Ayurveda, to go for the botanical drugs as well. Thank you so much.
Yes, agreed. No, agreed. I will make one comment on this botanical drug. If this FDA approves a botanical drug, there are, I think, couple of drugs got approved. There is something called equivalent to that in India. We have phytopharmaceuticals. So phytopharmaceutical, that is exactly equivalent to botanical drugs, where we need to identify 4 marker compounds. Then move forward. Basically, you need to establish that 4 marker compounds. You need to do pharmacokinetics, and also some of the activity also we have to show with at least 1 compound. That is a new approach recently, about 5-6 years ago, our DCGI in India. So it is really encouraging. And in fact, we run some of the programs, particularly IIIM and CDRI is leading some of those programs. So we are aware of that one. We are getting into that one.
Thank you so much. I hope to meet you sometime. I'll be in Hyderabad sometime in July. I hope to meet you. Yes, thank you so much. Yes.
It may be very late in the night for you.
No. Ours is now afternoon. Actually, it's afternoon tea time. It's very convenient.
Oh, you're in Japan, okay.
Yeah, they are 3.5 hours ahead of India.
Yes.
So not.
Yes. Yes. Thank you.
Anybody
Thank you
else
so
has any
much.
question? Anybody else has any question? Srinivas, would you like to make some comments on biotransformation?
Can I ask a small doubt with Professor Reddy?
Yeah, please.
Who is
Harish from Japan.
Yeah, go ahead, please. Go ahead, Harish.
Thank you, Professor Reddy. It's an excellent talk. I would like to know your thought process on the botanical drug, how it can be converted in terms of effective clinical dose when it comes to the human trials, sir.
Say it again, question.
Sir, in the botanical drug, effective clinical dose is the important for the human clinical trials, sir. If all those active metabolites from the plants may be having some microgram or nanogram level per kg of the crude extract or whatever it is, crude extract or crude per gram dry weight basis. If it comes to the human trial, effective clinical dose, how it can be managed, sir?
So basically, we need to make multiple batches, and we need to see that the reproducibility of this one. There is a fingerprint region. Also, we need to see, and we need to measure particularly these 4 markers. What we are looking at, particularly phytopharmaceuticals. So their range you need to define. You may not be able to define exact quantity, but you need to mention the range. This compound A ranges from 0.2 to 2.5%. That kind of ranges you need to mention. So these you need to be maintained in that form. Then again, you need to go to any. You can use the same as what NC approach what we use for single compound.
Okay.
Did I answer your question?
Yes, sir. Yes, got it. So thank you.
Anybody else has any question or comment?
Sure.
I was asking. Sorry. Yes, please.
Ask him to be part of your meeting in Trivandrum this year so that we can ask him more questions.
Yes. That will ensure. Srinivas has kindly agreed that he will try his best to come to Trivandrum to attend this event this year.
Because there are many questions and many comments. Cannot finish them here today. We need him there.
I would love to get connected with all seniors like you, sir. Definitely.
Srinivas taking the matter as mentioned by Professor Singh, that normally one question I ask, I was asking about transformation, but I leave that behind and maybe we'll discuss it at some other platform. One question for the benefit of members of society or all those who are attending this, lecture, which is also being telecasted live on YouTube, that you have, contributed significantly what you call good science. And also you have been very conscious about translational research. You brought a good juncture between the both two. And in the beginning and during your presentation and at the end also, you very clearly emphasized on collaboration and network, which is one of the very strong component in our society. So my question is, or point is that what would you like to suggest to today's researcher broadly who are attending this lecture, or who would be again looking at YouTube lecture which will be available, and also to the community of CSR scientists in particular in today's time when there is a lot of changes. At least we hear that which direction and how they should present themselves in order to succeed individually as well as a team for growth of science and technological avenues in this country.
Thank you, sir. Again, I echo. I think that collaboration is the only way forward. I don't think without collaboration, you may do small projects at individual level. If you want to achieve something big, I think collaboration is the only key. And particularly scientists who are working in like CSR labs like us, I think application-oriented synthesis, application-oriented science is very, very important. And I always tell my colleagues or some of the colleagues whom I interacted earlier also, always have a balance between basic science and applied science. Always have balance. Basically, basic science will give you publications and all. I mean, like kind of creates knowledge that will last longer. And in case of applied science, immediately you may not get benefit, but that is a way forward. Particularly CSR scientist, I can tell you that I always recommend both balance out basic and applied. And I particularly after working in industry and abroad, I think that collaboration is the only way forward. And probably, although just I'll give you my own example, not that I'm talking high about me, but see, we know only in our group, we only know how to make molecules. So after making molecules, what next? I can get published and I can get away. But ultimately, what we wanted to see that take forward towards application oriented, that's where I get connected with all the biologists and collaborations. Even if you look at, I didn't talk about some of the other work, particularly in mosquito repellents and crop protection, pheromones, some of the work what we have done. Without collaboration, I don't think we would have done some of the work what we can claim today. So that's where I think collaboration is the way forward. I think days of working alone, I think that's gone. I think future is about collaborations. I'm sure BRSI is in the direction and it is in good hands. And I'll be happy to support from my side in whatever capacity in taking forward this BRSI and also connecting with people and moving forward. Thank you, sir.
Thank you, Srinivas, that we appreciate a lot your contribution to the science, and we value your time given to us for this lecture. I request before I will say final, okay, Vinod has appeared here. Over to you, Vinod. Question session is over. And
okay, thank you. Thank you.
Professor Khare is also there.
Yeah. Thank you, Dr. Srinivasa Reddy, for this excellent talk. The lot of things we could able to learn about the natural product the total synthesis and the drug discovery approach. Thank you once again for taking time to deliver your lecture in this BRSI Global Lecture Series. Thank you very much. And I also thank Professor T.P. Singh, Professor Khare, Professor Ashok Pandey, and all other members who joined in this event. And thank you very much. And we will meet in the next lecture series, which will be in the month of June.
Thank you. Thank you all.
July. Yes, July.
Thanks, everyone.
Thank you. Thank you all. Thank you. Bye-bye.
Thank you, everybody, for a very nice talk. Thank you, Srinivas.
Thank you.
And thank you, Ashok.
Thank you.
Thank you.
Thank you all. Thank you all.
Everybody
Thank you.
else.
Bye.